Good afternoon. I'm Prem Paul, Vice Chancellor for Research and Dean of Graduate Studies, and I welcome you and thank you for joining us today as we convene this Nebraska lecture, our second this year in the Chancellor's Distinguished Lecture Series. This lecture is being held in conjunction with our 5th annual research fair. I hope you've taken the opportunity to hear some of the outstanding presentations given earlier today. Be sure to attend tomorrow's presentations. And first presentation tomorrow is early, 8 o'clock, by Dr. Nathaniel Pitts, who is the Director of Integrative Activities, National Science Foundation, and under his jurisdiction, EPSCoR program now houses. There'll be graduate student poster competition, and of course don't miss the undergraduate poster session on Friday. The Nebraska lectures are sponsored by the UNL Research Council in cooperation with the Office of the Chancellor and the Office of the Research and Graduate Studies. The Research Council, composed of faculty from across all disciplines at the University of Nebraska-Lincoln, supports a number of competitive awards and programs designed to encourage and enhance research, creative, and scholarly activities for all members of the UNL family. The Nebraska Lectures are one such program. The Nebraska Lectures are an interdisciplinary lecture series designed to foster communication among students and faculty in different academic areas and among citizens of Lincoln and Nebraska. The Research Council chooses Nebraska lecturers from UNL faculty on the basis of major recent accomplishments and the lecturer's ability to communicate his or her work to a multidisciplinary audience. I'm pleased to inform you that today's lecture is being web-streamed live around the globe. Dr. Dana Fritz, Associate Professor of Art and Art History, is Chair of the Research Council. I invite her and other members of the Research Council to please stand and be acknowledged. Would you please? Following Professor Larkins' lecture, Dr. Alan Christensen, Associate Professor of Biological Sciences, will moderate the question and answer session before we move to the reception in the Regency A Suite, which is located just across the hall. Typically, Chancellor Perlman formally introduces our speaker. However, due to the death of his mother, he is unable to be with us. In filling in for him, I'm pleased to now introduce Dr. Larkins. Our speaker joined UNL's faculty just 2 weeks ago, but he is no stranger to the institution or the state. Dr. Brian Larkins is a graduate of UNL and is a highly talented scientist. He joined UNL in March as Associate Vice Chancellor for Research, and he has academic home in agronomy and horticulture and School of Biological Sciences, and also holds John F. Davidson, Ph.D., and Marion J. Fuller, Ph.D., Chair in Life Sciences. A member of the National Academy of Sciences, he is internationally known for pioneering research on the nutritional quality of corn and other cereal grains. His research has implications for improving human nutrition, especially in developing countries where corn is a dietary staple. He has been a leader in the development of plant molecular biology and plant agricultural biotechnology. His portfolio includes advising the relevant unit on a unified strategic plan, enhancing the life sciences research, and mentoring and advising younger life science faculty. His appointment in agronomy and horticulture, as well as in School of Biological Sciences and in the research office, creates for UNL a unique opportunity to engage a person with outstanding credentials who also understands the traditions of this university. Perhaps most significant of all among his many achievements is that, like Chancellor Perlman, he's a graduate of York High School and of this institution. While we have assigned him an important task, that may resemble herding the proverbial cats, we also decided that his first assignment would be to deliver this Chancellor's lecture. With his considerable background in improving the nutrition of cereal grains, Dr. Larkins is uniquely suited to explaining to us the difficult task of how the world will feed its growing population as we are challenged by loss of arable land, global climate change, and water restrictions. The title of his lecture is Feeding 9 Billion People and Other Challenges to Agriculture. Please join me in welcoming Dr. Brian Larkins.
Thank you, Prem, and thank you all for coming out on such a cold, wet afternoon. I was asked to tell you about my research, what I do, and how I came to do it. And so I thought I would start by putting it in a context that I've been working in for the last 30 years, which has to do with creating more nutritional food for humans and for our livestock. So let me begin by just showing you a rather striking graph. This shows you here in the bottom left-hand corner the rate of growth of the human population. It's sort of what we call a hockey stick graph. You can see that we've been doing really quite well in this regard. It's estimated that about 2,000 years ago, the human population was about 250 million people. It took about 2,000 years to reach 1 billion people. It only took about 100 years to reach 3 billion people. And it only took about another 25 years to reach 5 billion people. So we've already passed 6 billion. And they estimate that the population is growing at the rate of about 180 million people a year. So if everything goes as planned, we should pass 9 billion people within about another 40 years. Now, for many of the people in this audience, I mean, this is not a far-off thing or a wild prediction. This is something that will happen in the lifetime of many of the people who are in this audience. And if not in your lifetime, certainly in the lifetime of your children. So what does that mean? Well, it's very simple that in the next 50 years, we're going to need to produce more food than in the whole of human history. Furthermore, we're going to have to do this on probably less arable land than we've had before. So it's very clear from this that we are going to have to find ways to increase crop productivity. Now, this first quote at the top comes from a recent United Nations report that came out just a month or so ago. They estimate that 18,000 children die every day because of hunger, and 850 million people go to bed at night with empty stomachs. Now, that sounds like starvation to me, but the real general issue is not so much one of the absence of food as it is a chronic undernutrition where people, especially children, do not get the proteins and the calories that they need in order to develop normally. So it's estimated that if prevailing trends continue, for every 100 children born, 94 will live to age 1, 91 of these will live to age 5, and of these 91, 28 will be malnourished. It's about a third. Now, most children that suffer from poor nutrition die of diarrheal diseases. But it's very clear that there are perhaps equally serious problems with deficiencies of micronutrients, especially iron, iodine, and vitamin A. So chronic deficiency of iron can lead to lethargy, but over a period of time it also leads to retarded mental development, and so these children have disabilities that can persist throughout their life. So this is a map that shows you hunger across the globe. And to make this really simple, if you look at the United States and you look at Europe, you don't really see any problems at all. The problems are in Asia and Africa and South America. So virtually all of the world's population growth, these billions of people, are going to come in these low-income countries of Asia and Africa and Latin America. And most of this will be in nations whose agricultural resources are already fairly limited. Now, the bottom line is, although these areas of population growth are regionally limited, the health, political, and environmental problems of these areas will be shared with all of the rest of the world. So it's not that we can simply disregard this as somebody else's problem. So these are three questions that come from Vaclav Smil's book, which came out several years ago, called Feeding the World, A Challenge for the 21st Century. So the first question is obvious. Can we continue producing enough food for the population which may undergo yet another doubling in the next 2 generations? Secondly, can we satisfy not only the basic metabolic requirements, but also the demand that will be generated by rising disposable incomes? So as incomes rise, people's demand for better diets also changes, and there's a greater demand for more meat, which means a greater demand for cereal grains to produce those livestock. Finally, can we do this without greatly increasing the extent and the intensity of undesirable environmental changes, without compromising many of the irreplaceable biospheric services? So, about coincidentally with Smil's book was a report from the National Research Council called Our Common Journey, A Transition Towards Sustainability. So the issue of a sustainable agriculture and a sustainable way of living is really an issue that affects all nations of the globe. So the goal of this sustainability is to basically meet the needs of a larger but stabilizing human population. It's estimated the population will stabilize somewhere between 9 and 10 billion people. To sustain the life support systems of the planet, and finally to substantially reduce hunger. I don't have time to give you all the details of this report, but basically these issues of sustainability have to do with providing food and nutrition, nurturing children, finding shelter, providing an education, finding employment, ensuring the quality and supply of fresh water, controlling emissions of gases into the atmosphere, greenhouse gases, and fluorinated hydrocarbons that destroy the ozone, protecting the oceans from pollution, maintaining species and ecosystems, and reducing hunger and poverty. So, we can ask ourselves, is the way in which we practice agriculture in this country sustainable? Well, our agricultural system has evolved over the last 100 years pretty much based upon these 5 things: plant breeding, mechanization, irrigation, the use of fertilizers, and pesticides and herbicides. And to say the least, that it's been highly successful. If you look at the yield of corn in the United States and the yield of soybeans in the United States over the last 20 years, they've gone up dramatically, although they are beginning to plateau. But what does it cost us? So the negative impacts of these annual crops that we grow takes its toll in terms of inefficient use of water and nutrients and soil loss. It's estimated that in the U.S. we lose 2 billion tons of topsoil a year to erosion, and the cost of this in lost productivity and silting and pollution of waterways is on the order of about $40 billion a year. No one ever measures those costs. And I don't know if you're familiar with Wes Jackson and the Land Institute in Salina, Kansas. If you've never heard of this, it's a very interesting concept. Wes is trying to develop perennial crops rather than annual crops. Wes's take is that humans made the biggest mistake they ever made 10,000 years ago when they domesticated wheat, rice, and corn, because it's by growing those annual crops that we create all these environmental problems for ourselves. Anyway, he estimates that of the 400 million tillable acres in the U.S., 350 of these range from mildly to highly erodible. So, the challenges that we really face in the next 10 to 15 years, and I say 10 to 15 because we've got to have this figured out before 40 years from now, or we're really going to be in a pickle, is how to increase crop yields to provide more food and maintain more open space. How to reduce inputs, that is, water and fertilizer for growing these crops. How to reduce environmental impacts of intensive agriculture. How to optimize these crops for human health and nutrition so that what we do produce can go a lot further than it currently does. And finally, to facilitate the conversion from a petroleum-based energy system to a plant-based renewable energy system. And of course, I'm sure you're all familiar with this final thing since it's kind of a hot item in Nebraska. So, agricultural research has changed a lot in the last 30 years. What I would call modern agricultural research still relies on these 5 things that I described to you before. These are really the arms and the legs of production agriculture, and they have to be included in anything that we do. But research in agriculture has changed a lot in the last 30 years. It's certainly become far more molecular and cellular, and it's led to the use of DNA markers for plant and animal breeding, clone genes that are introduced by transformation to create novel traits in crop plants, and basically defining how these valuable traits work by doing biochemical and genetic analysis of these particular traits. And I'm going to give you some examples of these in a bit. So this is a table that shows you a number of traits that have been introduced into crops in recent years. Most of these are at least in field trials, but a number of these that I've highlighted are actually in production. Clearly, I haven't time to talk about all of these, but the ones that really have been the most successful and the most widely used are herbicide tolerance, virus resistance, and insect resistance. And I'll just give you some examples of those. So I'm sure that people in Nebraska are quite familiar with Roundup Ready soybeans. Now, I think the estimate is that we use about 80% of the soybeans that we grow are genetically engineered for resistance to Roundup. And it's had a huge impact on farming in terms of reduced fuel use and reduced soil erosion. So the other major trait is insect resistance, which comes from transferring a gene from this bacteria, Bacillus thuringiensis, it's generally known as Bt toxin, into a number of crop plants, notably cotton and corn. In Arizona, where I've spent the last 20 years, it's very, very important. This graph shows you the reduction in the application of pesticides for growing cotton. And you can see that it's really a dramatic decrease. In fact, it's a reduction of millions and millions of pounds of pesticides that are no longer sprayed into the air between Tucson and Phoenix. And this is resistance to a virus called Papaya ring spot virus. So this is a field in Hawaii, and what you see here are trees that have been basically wiped out by the virus, and these trees were genetically engineered to be resistant to that virus. In fact, the whole papaya industry in Hawaii was virtually wiped out, and it was only through the research of one individual that made these transgenic plants that the industry has come back. So this is rather old data, but just to give you an example of data from 2001, it's estimated that these herbicide-resistant crops, mostly soybeans, saved over 234 million gallons of fuel, over 247 million tons of topsoil was conserved and protected from erosion because of reduced tilling, and 8 million tons of pesticides were not sprayed into the atmosphere. So clearly, this is something that everyone has benefited from, besides just the farmers that use this technology. So, this is where I come in. So, back in the early 1970s, I was a graduate student here in the Department of Botany, working with Eric Davis on a problem to try, a question to try and understand how the apical meristem of pea plants grew, how these cells enlarged. And the hypothesis that we were working on was that the plant hormone auxin regulates the expression of a gene that encodes an enzyme called cellulase. The walls of plants are made out of cellulose, and cellulase, we thought, might clip those cellulose fibrils, allowing the cells to expand. So the question I was trying to answer, or trying to demonstrate, was that the application of this hormone caused the activation of this gene. Now, to do that, I needed to be able to show that there was more cellulase messenger RNA produced. And so that required that I be able to isolate intact polyribosomes. So, since this is a general audience, I'll give you a little primer on polyribosomes. So, as I'm sure you're all aware, the nucleus of the cell contains DNA, which has the genetic code for making all of the enzymes and proteins that the plant is going to use throughout its life. Now, that information is copied from the DNA in the form of a messenger RNA, which goes out into the cell, and it is read by ribosomes. And basically, these ribosomes read the nucleotide sequence of the message and convert that into an amino acid sequence, which is the protein. Now, a polyribosome is basically a long messenger RNA that has a number of ribosomes that are reading it. So you can see here a number of ribosomes along the message, and you can see proteins that are coming off of those ribosomes. So that's basically what a polyribosome is. Now, I have to tell you that we never did totally answer the question about what auxin does to cellulase, but there were two really important things that came out of this research. And I say this somewhat modestly, but I've been told this is true. First of all, this research provided a means to isolate messenger RNAs and genes from plants and study their expression, which up until that time had not been possible. The second thing was that it inspired a number of scientists to use molecular genetic research to study how plants grow and develop, and to develop methods for crop improvement. So, this is the next transition of my life. Around 1964, there were a couple of scientists at Purdue University, Ed Mertz and Oliver Nelson, who discovered a mutant in corn called opaque-2. And this generated a great deal of excitement. So why? So corn, like most cereals, including wheat and rice, has a fairly high content of protein, so usually around 10% or so. But the protein that it contains is deficient in one or more amino acids that we cannot make for ourselves, or what we call essential amino acids. The most limiting of these amino acids is lysine. So what Mertz and Nelson showed was that this mutant almost doubled the lysine content of corn. And it generated a lot of excitement at the time. They thought it would turn out to be a panacea that would solve the world's protein deficiency problems. So, in fact, on the left here you see a pair of litter mates, one which was fed corn from opaque-2 and the other which was fed wild-type corn. On the right you see a child. This is from a study in Guatemala. This child was suffering from a protein deficiency disorder called kwashiorkor, and after several months of feeding primarily the corn-based diet, that child recovered from kwashiorkor. So obviously this is nutritionally far superior to normal corn. Unfortunately, this mutant has several limitations. So the first one is that opaque-2 endosperm, as you can see here, is soft and chalky. Now, what that means is it makes it more vulnerable to insect damage, and these kernels tend to break easily when you're harvesting them. And perhaps its most damning thing is that it makes really lousy tortillas and tacos. So the people who would have benefited the most from this didn't want to use it. Finally, opaque-2 is a recessive mutation. Now, what that means is if you have a field of opaque-2 corn growing in the vicinity of a field of normal corn, and that normal pollen blows onto that corn, then you lose the effect of opaque-2. So it clearly had some problems, and actually within about 6 years of the discovery, most of the air went out of the balloon. However, plant breeders at the International Maize and Sorghum Improvement Center in Mexico City, which is generally known as CIMMYT, discovered that when you cross this mutation in certain types of corn, that instead of getting soft, starchy kernels, that you got normal kernels. And they called this quality protein maize to distinguish it from this typical soft type of corn. Now, quality protein maize, which is called QPM, is basically just like normal corn. The yield is pretty much similar to wild type corn, and the protein quality and digestibility, though, are as good as what you get with opaque 2. And so, in fact, this QPM corn is currently being developed in China and Africa and South America. But it's difficult to do this because in order to convert local germplasm into QPM germplasm, you have to monitor 3 things. You have to monitor protein quality, you have to monitor these opaque 2 modifiers, and you have to maintain this recessive mutation. So it does take a little bit more effort than what most plant breeders in companies here in the U.S. are willing to put in. So this leads me then to the 2 questions that we've been basically trying to answer. The first one is, what mechanism does opaque 2, or by what mechanism does opaque 2 increase the kernel lysine content? And the second question is, what is it that determines kernel hardness? Now, you might find this surprising. People have been growing corn for centuries, but we really don't know what makes it hard. So let me tell you a little bit about corn. You probably eat it, but you've never dissected it before. So this is a longitudinal section of a mature corn kernel. And most of what you see here at the top is what we call endosperm tissue. You can see that the central part is white and starchy, and the margins are dark and yellow. This is what we call the vitreous endosperm. It's the part that makes it hard. And when they dry mill corn, this is what they make cornflakes and grits out of. So it's a pretty important part of the kernel. So the endosperm is where you find the starch and most of the protein. So a kernel is typically 8 to 10% protein, and about 90% of the protein is found in the endosperm. 70% of that protein is a storage protein that is called zein, which I'll tell you about in a second. And zein basically has no lysine in it. Now, what you see at the bottom then is the embryo, and this is where the oil is stored, and that's all I'm going to tell you about the embryo. So this is a scanning electron micrograph of a developing endosperm cell. And if you look, you can kind of see the walls around this cell. You'll see these large balls in the middle, which are starch grains, and this white fluffy material is the endomembrane system. And in that, you'll see these little round balls, which are called protein bodies, and these are the sites where this storage protein accumulates. Now, if you make a thin section through the cell, here you see in higher magnification what those protein bodies look like. If you extract those proteins from the protein body and separate them by size, you can see that there's a number of different proteins in there. Over a period of years, we isolated the genes that encode each of these proteins, so we know exactly what their amino acid composition is. And I can tell you that there's basically no lysine in these proteins. And as I said, since there's 70% of the protein in the seed, and there's no lysine in it, you can understand why the lysine content is low. Now, this is an unusual opaque 2 mutation. This mutant is as a result of a transposable element, or a so-called jumping gene. And when this transposon jumps out of the opaque 2 gene, it leaves behind a normal gene. So if you look at this section on the right-hand side, these cells at the top are normal cells, and the cells at the bottom are mutant cells. And if you look carefully, you can see that there's starch grains and these little dark staining protein bodies in the normal cells. But here in the mutant cells, it's very hard to see protein bodies because of the consequence of this mutation. Now, if you were to extract the protein out of those cells, on the left you see the zein proteins that come from the normal genotype, and in the lane next to that you see the zein proteins that come from that opaque 2 mutant. Key thing is that you can see that some of these bands are greatly reduced in the mutant. So one of the effects of this mutation is that you make less zein proteins, and if you can see this over on the right-hand side, the upper cell has normal protein bodies, and the lower cell has much smaller protein bodies. This is a mutant cell. So the other thing that happens is if you look at the other proteins in the cell, The one on the left is, again, the normal one, and the one on the right is the mutant one. And you can just see from the way that these proteins stain that there's many more proteins here in the mutant than there is in the wild type. So that's why the lysine content goes up. So in this table, you can see on the left a normal genotype, an opaque 1 mutant, which is a different mutant, and the opaque 2 mutant. So in the normal genotype, there's about 12 milligrams of protein, also about 12 milligrams in this opaque 1 mutant. There's about 8.2 milligrams of zein protein in these two, and about 2 milligrams of non-zein protein, and the lysine content ends up to be about 1.5%. In opaque 2, there's less total protein. There's a big reduction in zein proteins and a big increase in non-zein proteins, and you end up almost doubling the lysine content. I should tell you that what is required for humans and most other monogastric animals like pigs and chickens is something on the order of about 5% lysine. So opaque 2 is quite an enhancement, but it's not perfect either. Okay, so by what mechanism does opaque 2 increase the lysine content? Well, in 1990, Bob Schmidt, working in Ben Burr's lab at Brookhaven, isolated the gene that encodes this opaque 2 protein. And it turned out to be a transcription factor. Now, transcription factor is a protein, which you see here, which binds to regulatory regions of genes and turns them on. It's like turning a switch to make the gene work. So when this protein is there, the genes turn on. When the protein is not there, the genes never turn on. Now, we used what's called a microarray hybridization. Basically, each of these little dots that you see represents a different gene, and this is just part of the overall study. But we surveyed the expression of all of the genes that are expressed in the endosperm to identify which genes are affected by this mutation. And this table tells you what they are, which is probably hard to see, and it's really not that important. The main thing I wanted to point out is that we found that over 250 different genes are affected by this mutation. So you can see that it's what we call a very pleiotropic mutation. It affects many things. Okay. Now, I guess I should also tell you that we did identify a protein that was affected that goes up in this mutant. This protein contains about 10% lysine, and this protein turned out to be highly predictive of the lysine content of the grain. Basically, you can use the level of that protein as a selection tool to select genotypes that have higher lysine contents. But this leads me to our second question, which is, what determines the hardness of the kernel? And you can really divide this into two questions. The first one is, what's the biochemical property of endosperm cells that makes this vitreous phenotype? And secondly, by what mechanism do opaque 2 modifiers restore a vitreous phenotype in these QPM lines? So we used 3 different experimental approaches to try and answer this question. The first one is to characterize mutations that cause this opaque endosperm phenotype. There's a number of these that are known, but no one knew exactly what the nature of those genes is. Secondly, we did a biochemical analysis of O2 endosperm, of modified O2 endosperm, to try to find out what made it different than normal endosperm or opaque endosperm. And finally, we began an experiment to genetically map these modifier genes with the ultimate aim of cloning and isolating these genes. So I don't have time to tell you all the details of this. I'll just tell you a little bit about it. There are actually about 20 mutations that cause this opaque phenotype. And in theory, some of these could be these opaque 2 modifier loci, because if mutated, they would end up giving you a starchy endosperm. So this is a table showing you or a figure showing you the 10 Maize chromosomes. And these colored bars are where zein genes are found. And I've highlighted here different mutations that give rise to an opaque phenotype. Now, over the years, we isolated 6 genes that cause this phenotype. And I won't tell you about very many of these, but 3 of these genes turned out to be mutations in the zein proteins themselves. So what you see here at the top are these normal protein bodies, which I showed you before, and you'll notice that they're nice and round spherical structures. What happens in two of these mutants, Flowery2 and DEB30, you'll notice that the protein bodies are much smaller and they're sort of glued together. So the consequence of changing a single amino acid in these proteins causes the proteins to misfold, and it creates a syndrome in these cells that's not unlike the syndrome that you find in the neurons of people who suffer from Parkinson's and Alzheimer's disease. So there's a lot of similarities between plant cells and animal cells. I can tell you some more about that later if you're curious. Okay, so in the course of looking at the biochemistry of modified opaque 2 endosperm, we found something really surprising. And that was that the starch from these modified opaques swells more than normal starch. So here you see starch from a normal genotype, in the middle opaque 2, and you'll notice these pellets are about the same size, whereas the one on the right, it's nearly double in size. So this turned out to result from a difference in the structure of one of the components of the starch. And this leads to different properties of the kernel when the thing dries down. So what you see here on the left is a scanning electron micrograph of a mature, normal kernel in that vitreous area. So again, you see the starch grains, and you'll notice these, you can see protein bodies are sort of packed around this. And you can also see some rather nondescript material, which is just dried up cytoplasmic material. So in a normal kernel, when it matures and it loses water, basically all the contents of those cells congeal together. And you end up with having protein bodies and dried cytoplasmic material that packs around the starch grains. So this creates a matrix that will transmit light. What happens in the opaque mutant, on the other hand, you see over here, you don't see many of those protein bodies or any of that matrix. And so you have air spaces between the starch grains. So when that kernel is put in the light just bounces off of the starch grains and it appears to be opaque. Now, these modified opaques were different. If you look at these starch grains, you'll notice there are these little compression faces on the surface of these starch grains. And basically this results because these starch grains bind more water. So as the kernel matures and it dries down, those starch grains compress together. So you end up making another type of matrix in this kernel of the starch grains just being glued together, and that's what makes them translucent and potentially harder. So we have two hypotheses then about factors that affect the hardness or the vitreousness of the kernel. The first is that it has to do with the structure and the number of these protein bodies that pack around the starch grains. And the second one is that it has to do with the structure of the starch itself. So you could have two quite distinct biochemical mechanisms that influence whether or not you get a hard corn kernel. Now, our next step then is to try and isolate those genes and answer that question. And to do this, we started off with a modified opaque kernel, which you see here at the top, and we crossed that to a soft opaque kernel, and the F1 hybrid is intermediate between the two. If you plant this seed and self-pollinate the ear, then you get an F2 ear that segregates, and you get some kernels that are vitreous and some kernels that are opaque and everything in between. So it's a very complex trait. Now, if you were to plant one of these kernels and continue to self-pollinate this for 6 or 7 generations, you genetically fix the genotype of each one of these kernels, and it produces what we call recombinant inbred lines. Now, we've used DNA markers to map these modifiers, or what we call quantitative trait loci, because there are multiple loci affecting this trait. So this is the distribution of these DNA markers on the 10 chromosomes. And we can look for the co-segregation of this modified phenotype with these markers. And basically, we've identified 5 loci, 2 here on chromosome 7, 2 on chromosome 1, and 1 over here on chromosome 9. And in fact, this modifier here on the short arm of chromosome 7 is a really major modifier. It accounts for 40 to 50% of the effect. So it's what we call a major QTL. Now, we're in the process of backcrossing these individuals to the original soft parent, which will allow us to identify which segments of the DNA contain these genes. And it just so happens that by the end of next year, we will know the complete nucleotide sequence of the corn genome. And so it should be fairly straightforward to walk to these loci and identify what these genes are. And I should tell you that there's lots of scientists in this country, in fact around the world right now, who are doing similar kinds of experiments mapping genes with the idea that once we have the knowledge of the nucleotide sequence of this genome, we're going to be able to figure out a lot of things about what makes corn work. Okay, one last thing I'd like to tell you about, and this is not our work, but this has to do with some work that has been done at Monsanto. So there is a new kind of mutation that you can make. It's called RNAi interference, or RNAi. And basically to do this, you take a gene and you snip a little piece of it out, and then you turn that around and you put it back in. And the consequence of this is that when it makes a messenger RNA, that messenger RNA has a structure which the cell recognizes as being foreign, and it destroys it. So you can take the genes that encode these different Z proteins that I've told you, and you can make these RNAi structures and basically destroy the messenger RNAs for making Zeins. Now remember, I told you Zeins have no lysine in them. Now, I don't know how well you can see this, but on the left-hand side you see one of these RNAi mutants, and on the right-hand side you see the normal corn. So you'll see that these RNAi mutants are, guess what? Opaque, right? Now, this table shows you the amino acid composition of these mutants. So at the top you see the wild type, and here on the left are the amino acids. And I just point out two of these, lysine and tryptophan. So these are in parts per million. So this wild type is 2.5% lysine and about 0.5% tryptophan. These four genotypes are transgenic lines where they've knocked out the synthesis of Z proteins. And you can see that they range from 4 to 5% lysine. In other words, the protein in these kernels would be comparable to the protein in milk. Now, we're in the process of testing if these modifier genes that we've been working on can modify these RNAi mutants, and our initial results are very promising. So we think that we should be able to put these modifiers into these RNAi mutants and create a corn that was what I had set off to do about 30-some years ago. Anyway, and I really think that realistically you can expect this could happen within the next 2 to 3 years. This would also have immense value for those of you who might be interested in producing alcohol from corn, because you would end up with a protein product left over that would be much more valuable for feeding livestock. Okay, so let me just summarize what I've tried to tell you. So our research has described the nature and the mechanism of synthesis of the major storage proteins in maize. I sort of glossed over that, but it did take us about 15 years to get that all worked out. Quality protein maize is high yielding and it provides a nutritious source of protein for monogastric animals. But creating QPM germplasm is genetically complex. And if we can identify these modifier genes, then this becomes a whole lot simpler and a lot faster. Finally, there's immense potential for further improving protein quality in corn through genetic engineering with these RNAi mutants. So hopefully that's something that you'll see in the next couple of years. Now, there are people in this university and the universities across the country and in companies that are working on a number of traits that would dramatically improve the foods that we produce. There are people who are trying to remove allergens from peanuts. They've already removed the allergen from soybeans. I told you about our work on increased protein quality, trying to raise iron content, high level of omega-3 oils, which is good for your heart, increased vitamins, increased antioxidants will let you live an extra 20 years, cholesterol inhibiting substances, and so on. So I don't have time to describe all that to you, but I would like to tell you a little bit about some of the work that's going on here. So I can only give you a couple examples of this, but this comes from Tom Clementi's lab in the Department of Agronomy and Horticulture. He's worked out a method to transform genes into all of the major crops that are grown in Nebraska: maize, wheat, sorghum, soybean. His particular research is related to genetically engineering soybean oil content. He's trying to increase the level of oleic acid. So this could be used as a biodiesel. So I'll leave it to Tom to explain to you about how much progress he's made on that. Now, Don Weeks in the Department of Biochemistry a few years ago isolated a gene from a single-celled algae that grows in ponds in Nebraska. And this gene produces an enzyme that is able to destroy this herbicide called Dicamba. And working with Tom Clemente, they transferred that gene into soybeans. So here you see soybeans that are transgenic and soybeans that are not. So this has really immense potential as the next generation of a herbicide-resistant crop plant in addition to Roundup. So finally, Sally McKenzie's lab is using this RNAi technique that I described to you to create male sterile plants in tomato and soybean. So male sterility is used for making hybrids. And if you can do this in this fashion, you can create hybrids a lot more efficiently. So it's all pretty exciting. So how did I get here? Well, I grew up on a small farm outside of a town called Chester, which is in the southeast corner of Nebraska. Chester's not noted for a lot of things, but it is they did win 2 Class D basketball championships, and it also is the site of where six-man football was invented. Of course, that's not very important anymore. But in any case, that's where I got my start on this small farm. This is a picture of our farm that was taken about 1955. About 1956, these portions of the farm were removed by a tornado that came right down the middle of our driveway. This is a picture of me and a couple of my brothers and sisters and our neighbors. Here you see me. I'm obviously giving a lot of thought to what we're doing. So I was very fortunate when I was 16 to move to York, Nebraska. York had just an absolutely terrific high school. Excellent teachers. I tell you, I don't think you could get a better education than the education I got in York, Nebraska. I have to tell you that I didn't meet Harvey Perlman when I got there. Harvey had already left and was at the university. But I did meet his brother. His younger brother was a year behind me in school, and I played intramural basketball against his brother. It was very obvious from his jump shot that he came from an intellectually gifted family. So then I came to the University of Nebraska, and I wish I had more time to tell you about these 2 gentlemen. The guy on the left is John Davidson, who was a botany professor that I took 3 courses from, and it's really the reason I'm standing here today. Les Whipp was in the English department. I took a course in English poetry from Les Whipp. I have to tell you that I never became a poet. But what these 2 professors had in common were 2 things. First of all, their door was always open, and it was very easy to go and sit down and talk to them. And I made Les late for class multiple times because we'd get so engaged in our conversation before class started. But what they both did was they taught by what's called the inductive method, which is to say they didn't lecture. They asked questions, and they taught us to ask questions. And as Doc Davidson used to say, you don't remember the answers to my questions. You remember the answers to your questions. And, it's really true. Well, anyway, I'd just like to end this by telling you a couple of things. First of all, the University of Nebraska is really an invaluable resource to the people of this state. One of my brothers and sisters attended the University of Nebraska, and I'm sure we wouldn't have been able to do that if it hadn't been for the fact that it was really fairly inexpensive to go here. Secondly, that for those of you who are students, the education that you receive here is a gateway to doing things that you can't even imagine at this stage. When I graduated in 1969, the first time, I had borrowed money to pay my tuition and fees every year. And I must say I also had a few scholarships from Teachers College. But Anyway, I graduated with this crushing debt of $3,200. Now, that doesn't sound like very much to you, but my wife and I bought a house for $11,000. Anyway, my father-in-law told me at the time, he said, you know, that's the least amount of debt you'll have your whole life. Well, my father-in-law bought a new Chevy Impala in 1968 for about $3,200. And so I checked the other day on the cost of tuition and fees at the University of Nebraska. 4 years of college here is about the same as a new Impala. So, you know, people think it's awfully expensive, you know, but, you know, it's pretty much just what it always was. So let me end this then by acknowledging all the graduate students and postdocs in my lab over the last few years who really are the ones that did all the work that I told you about. And I'd be happy to answer any questions that you have.
Thank you, Brian, for that lecture. Brian will be happy to answer questions from the audience. Please speak up, and he'll repeat the question for the benefit of the podcast. Also, after the questions and answers, there is a reception across the hall in the agency suite.
Brian, what's the disadvantage to corn to loss of zein?
Okay, so the question was, what's the disadvantage to the corn plant of not making zein proteins? Well, it appears that one of the disadvantages is that you end up with an opaque kernel, which would make it softer and more susceptible to insects and so on. However, there doesn't appear to be any negative effects in terms of germination or anything else. You just end up making more of some other proteins. Yes? Okay, so the first part, the question was, would I tie the first part of my talk to the second part of my talk? So the first part of my talk was basically to try to make the case that we face a dramatically increased population growth, and we're going to have a lot more people to feed, and we need to do this in ways that are not going to destroy our ability to produce that food. So what that means is we need novel ways of producing food that we've never used before. We can't just keep doing it the same old way that we're doing it because there's limits to what you can do. So you can produce cereal grains, for example, that have higher contents of iron, that have higher protein quality, that are more nutritious. And so that basically it makes those crops go a lot further than what we're able to use right now. So, for example, what we do in this country for livestock feed for corn is that we ferment, we grow bacteria that produce lysine, and industrially that lysine is fed into the cornmeal to produce rations for chickens and pigs and so on. So it costs somewhere between $350 to $400 million a year for these lysine substitutes. Now that's something that we can do in this country without too much trouble. But if you want to try and do that in Africa or Southeast Asia, it's not really an option. So these higher feed quality grains would have immense value in those parts of the world that I described to you where we're going to see the largest part of the population growth. Yes?
Would you describe the unintended consequences of genetically modifying crops?
Okay, the question is, would I describe the unintended consequences of genetically modifying crops? Well, that's a complicated question. I guess it would depend upon what the genetic modification was that you were talking about. Certainly, for example, in these grains, that if you knock out these storage proteins, so you increase the balance of amino acids in them to make it more nutritional, I guess one of the unintended consequences is you end up with a grain that's softer. Okay, there's certainly some of these things that you don't know until you test it out. But after that, it just depends on what you're talking about. With this RNAi technology, all that you're basically doing is creating a mutation of a gene so that that gene doesn't work. For the last 70 years, we've been creating mutations of genes by chemical mutagenesis, where you take the seeds or the pollen and you treat them with a chemical that mutates the DNA, and then you screen the progeny that you produce for a mutation in the gene that you were looking for. And that's been widely used in horticulture and agronomy. Now, when you do that, you get what you're looking for, but you get a lot of mutations that you don't even have a clue about. So we've never worried about all those other mutations that you've created because they just simply breed whatever they produce. So with genetic engineering, you can surgically go in and knock out a single gene. So I guess I would argue that you have far less unintended consequences of making mutations that way or changing gene expression patterns that way than you do by chemical mutagenesis, which we've used for a long, long time. Yes? The question is QPM still a recessive mutation? The answer is yes, it is. So in these areas where they're trying to develop QPM, that's all they grow. It has to be in isolation from any other types of corn. Yes?
Is the ultimate goal to eradicate the less nutrition types of corn? And if so, what's the probability that will happen in time to accommodate the growing population?
So her question is the ultimate goal to eradicate normal corn, right? No, that's not the ultimate goal. Although if this was to be successful technology, it's not hard to imagine that farmers might grow large amounts of this on contract for chicken feed or hog feed or whatever. It might be the ideal thing to grow if you're interested in producing alcohol. So it would never totally replace normal corn because, well, for one thing, you couldn't make tortillas out of it. Yes?
You keep on talking about monogastric livestock producers.
Yeah.
Can you feed it to ruminants? And if you can, is that going to be a big advantage for livestock producers?
Okay, so her question was, I kept talking about lysine being important for monogastric animals. Those are animals that have just a single stomach, like humans and pigs and chickens, but it doesn't refer to cattle or so-called ruminants. So it certainly would still be absolutely good fodder, or not fodder, but good feed for ruminant animals as well. It's whether you'd get as big a kick as you would get for monogastrics, I'm not sure, but I'm sure it would be better. Yes?
Are there any significant plant breeding programs going on in the developed nations that are aimed at Africa and Asia, the places where hunger is a problem?
Okay, so her question is, are there any significant plant breeding programs, or do you want corn breeding or just plant breeding? Plant breeding programs that are directed specifically at developing countries. Well, there certainly are a number of plant breeders that work on crops that are important in developing countries. And I mean, close to my old institution over in Riverside, California, they've been breeding cowpeas for years and doing a lot of work with varieties that are grown in Africa. And at Texas A&M and no doubt at the University of Nebraska, they're working on sorghums that are grown there. So I would say that it's not a major effort, but there certainly are efforts that are out there, and it's not hard to imagine that with the emphasis that the Gates Foundation is now putting on developing crops for sub-Saharan Africa and Asia, that there might be a lot more of that would happen. Yeah.
The deployment of this technology it's happened within the United States pretty readily, certainly within your career, and the people in this room have been involved in that. But to solve the problem that you introduced today requires deployment throughout the world, and not all the world has bought into GM type technologies. So is it as much a technical
Okay. So the question was whether the implementation of genetically engineered crops throughout the world is really going to be a possibility because it hasn't been widely accepted, particularly in Europe and so on. Well, as you probably know, there's been a huge campaign of disinformation to frighten people about this technology and what it means. I sort of had the impression that over the last few years, maybe simply because the rhetoric had sort of died down a little bit, that it wasn't quite the issue that it had been. I mean, I find it really quite impressive, to say the least, that Brazil basically outlawed growing Roundup-ready soybeans so that they could ship them to Europe. And but in Argentina, they started growing these Roundup-ready soybeans, and it was so successful that those little beans walked right across the border into Brazil. And it took a couple of years, but the Brazilian government finally legalized the production of Roundup-ready soybeans. So, I'm sure there must be some downsides to this, but, it's awfully hard to knock something that 80 to 90% of the farmers think is a pretty doggone good product. And, just in the savings in erosion and gasoline, not having to cultivate, seems to me like a pretty good deal. Now, all that you can hope is, eventually success will build on success, and these things will come to be more widely accepted, or else consumers and citizens will be a little bit more inquisitive about the facts of the matter. It's not to say that, you shouldn't be concerned about genetically engineered traits, but I can tell you that they're the most highly regulated crop plants of anything that's ever been produced in the history of this country, or the world for that matter. They wouldn't be out there if they weren't safe. Yes?
With an increasing population, you mentioned that there is a need in the future, an increase in production of crops. On the other hand, there is also a demand for biofuels. Don't you see the potential conflict in the two?
Okay, so his question is, with an increasing population and a demand for food, isn't there going to be a real competition for these grains or whatever you would use for producing biofuels? Well, of course, the ultimate goal for the biofuels production is to not use food crops at all, but to use cellulosic crops, grasses, and so on, that we simply don't have the technology to do right now. On the other hand, it's fairly unlikely that that technology is going to be developed in the next 3 or 4 years, so we're stuck with using grains. So it becomes a question of whether you can ferment the starch to produce alcohol and recover the protein to be able to make a useful food for livestock. So, you might be able to leverage this in such a way that it isn't a total loss. The only thing I would say about that is, it may not be all that cost effective to produce fuels this way. But then, we're spending money right now in a very cost ineffective way on other activities that we're engaged in. So it isn't that bad of a loss. Yeah. So his question is are these crops that West Jackson is developing at the Land Institute going to require heavy irrigation in order for this to work? So basically, I'm not sure I can remember everything, but so Wes is growing perennial sorghum. He's trying to develop perennial corn and a number of other grasses that could be grown. The real problem I have with Wes's idea is that, you could make this into a sustainable agriculture, but you can't make it sustainable enough to feed all the people that you need to feed. Well, his argument is that, well, if we'd have done this in the first place, we wouldn't have all those people today to feed in the first place. But, it could still have its merits. I'm just not sure what the total productivity of these perennial crops would be. But it certainly would be better for the soil.
Okay, well, before we go across to the